Hello everyone, I hope everybody is doing well. My name is Shyaron Poudel and I'm doing my master's in molecular biosciences here in Arkansas State University. Currently working under Dr. Asela Wijeratne. He is my mentor and research advisor. Today I'm here for the poster presentation of one of my current research work, and the topic for my research is reduced representation by sulfide sequencing to detect condition-specific DNA methylation in soybean. Our lab, we specifically focus on soybean because it is one of the most important crop in the United States for its nutritional value, export to different other countries, and diverse uses from the food industry to the textile industry. However, due to various biotic and abiotic stresses like pathogen infection, temperature, radiation, and water, we lose tremendous amount of yield annually. During these stresses, plant goes through different genomic modification, and DNA methylation is one of them. DNA methylation can affect plant production, survival, and development as well. So we can define it as an epigenetic modification which occurs when an enzyme called methyltransferases adds methyl group in the fifth position carbon of cytosine. Cytosine is one of the building block of DNA, and in figure number 1 you can see how DNA methylation happens. DNA methylation can directly affect the gene expression by switching off the gene by inhibiting RNA polymerase, which is an important enzyme for DNA transcription, to promoter region of that gene. And in figure number 2 and 3, we can see how gene silencing happens due to the inhibition of transcription due to cytosine methylation in promoter region. So study of methylone profile of a plant gives us an insight to elevate stress tolerance and increase crop productivity even during adverse conditions. In our research, one of our objectives is to compare whole genome bisulfite sequencing method, which is our conventional method for methylome profiling, with reduced representation bisulfite sequencing method, which is a novel method for methylome profiling. Even though both of the methods uses bisulfite converted DNA fragments, whole genome bisulfite sequencing uses random fragmentation method and sequences whole genome, but RRBS uses specific endonuclease restriction enzymes for selective fragmentation of DNA, and only those fragments are used for sequencing. So these differences make RRBS more cost effective and also more specific compared to whole genome bisulfite sequencing. Our second objective is to optimize RRBS library preparation method. You could see in figure number 5 on the methods and method. Both methods are similar. However, for whole genome bisulfite sequencing method, random fragmentation can be done by using sonication, and for RRBS method, specific endonuclease restriction enzyme such as MSE1 it can be used to cleave the genomic DNA in specific sites. So during library preparation, adapter ligated DNA fragmentation fragments were selected accordingly to their sizes, and then bisulfite conversion was done where non-methylated cytosine were converted into uracils. So moving towards the results included in this poster are preliminary data which only includes one RBL sample. In figure number 6, you could see you could visualize the smear of DNA fragment size between 200 base pair to 450 base pair length in the gel after gel electrophoresis, which tells us that the MSE1 worked very well as a restriction enzyme for soybean genome. And after receiving the sequence result from the sequencing facilities, quality assessment of data was done using FASTA QC and Fred box plot method, where in figure number 8, you could see that the Fred score for our sequence is more than 20 up to 40, which indicates that the quality of the sequence is very good enough for the downstream steps like cut adapt to get rid of adapters, a Bismarck for methylome profiling, and methylkit for the final the data processing. So let's move towards other results we obtained after aligning, or we can say comparing our sequence with database online. In figure number 9, we can see that the high methylation was observed in cytosine guanine dinucleotides, followed by CHG, CHH, and CHN context, H being nucleotides other than cytosine. And in figure number 10, it shows that 75% of genes from our sequence method were matched with the database. And also in table number 1, we could see that the 59.6% of reads were aligned with the database. And with all these results, we can conclude that 75% gene mapped with the database. In overall, MSE1 was a good choice as a restriction enzyme, and in future we need wider size range of DNA fragment to include more genes. And in coming future, our goal is to compare these two sequencing methods and use different other restriction enzymes to make RRBS protocol more specific. And finally, we want to thank AState internal seed grant for the funding, and Arkansas State University and Arkansas Bioscience Institute for the support and facilities. Thank you very much for listening to me, and have a nice day.